Hello everyone and welcome to today's keynote presentation titled Mammalian RNAi Response to Virus Infection. This webinar is part of the 2021 Microbiology Virtual Week. And I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive and we encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, simply click on that Ask a Question box located on the far left of your screen. As a reminder, the presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Shou-Wei Ding, a distinguished professor in the Department of Microbiology and Plant Pathology at the Institute of Integrative Genome Biology at the University of California, Riverside. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Ding, welcome. You may now begin your presentation.
Hello, everyone. My name is Shou-Wei Ding, and I am a professor in the Department of Microbiology and Plant Pathology with University of California at Riverside. The title of my talk today is Mammalian RNAi Response to Virus Infection. A lot is known about antiviral immunity in mammals. In fact, the entire world is using the knowledge gained from virology and immunological research to get us out of the current COVID-19 pandemic, hopefully soon. And antiviral defense mechanism in mammal can be divided into adaptive immunity, innate immunity. Okay, so adaptive immunity is mediated by B cells and T cells. It has specificity and memory. However, adaptive immunity takes time to activate. Innate immunity, in contrast, can refer broadly to all the mechanisms that provide protection before the activation of adaptive immunity. Innate immunity includes those that initiated by pattern recognition receptors, such as toll-like receptor, RISC receptor, cGAS, and so on. And innate immunity mechanism often depend on the production and the signaling of interferons, leading to the induction of interferon-stimulated genes to activate a broad spectrum antiviral state. Today, I'm going to present recent findings from my lab and collaborators on a distinct mammalian antiviral defense mechanism that is directed by RNA interference, or RNAi. I'm sure all of you know about RNAi, and it includes two key steps, dicing and splicing. So in the first step, long double-strand RNA is diced by endoribonuclease dicer into small interferon RNAs, or siRNAs. In the second step, these siRNAs are loaded into RNA-induced silencing complex, or RISC, in which Argonaute protein is an essential component. siRNA are a product of dicer enzyme, and is a kind of very special form of RNA molecules that is actually a short RNA duplex with 5' phosphate and 2 nucleotide overhang at each end of the 3'. Okay, so RNAi pathways actually direct antiviral immunity in plants, fungi, nematode, and insects. For example, in fruit flies, viral double-stranded RNA replicative intermediate are processed into virus-derived siRNA or VSRNAs by dicer 2. And these VSRNAs are then loaded into ARGO2 RISC to mediate ARGO2 dependent virus clearance to suppress viral replication. Okay. To counter defense, successful viral infection depends on a viral suppressor of RNA expressed by the viral genome. So these viral suppressor of the RNA or VSR is essential for infection. Okay. So in Florhaus virus, what I have listed here, or FHV, is a positive-strand RNA virus, was the first animal virus shown to induce ARGO2 dependent antiviral RNA in Drosophila cells, and also the first virus to show shown to encode a viral suppressor of RNA, and that is the B2 protein. So studies on Florhaus virus infection in fruit flies provide the most of the knowledge on what we know about antiviral RNA in fruit flies. And also these studies define how the Florhaus virus B2 protein to suppress RNA as a double-stranded RNA binding protein to sequester long double-stranded RNA and prevent dicer processing into siRNAs. In 2016, we published a joint paper with Professor Kate Jeffries' lab, providing the first example for a human virus to induce and suppress antiviral RNA in mammalian cells, based on studies on influenza A virus. Influenza A virus needs no introduction. Just to mention that this virus contains a segmented negative strand RNA genome. The reason why one of the main reasons why we chose to work on influenza virus is because in 2004, we have published a paper showing that the non-structured protein 1 of influenza A virus is a suppressor of antiviral RNA in Drosophila cells. So to maximize detection of RNA response, we infected human cells with a NS1 deletion mutant of influenza A virus the PR8 strain, kindly provided by Dr. Adolfo Garcia Sastre. This slide, the top panel of this slide shows the northern blood detection of viral siRNAs from monkey viral cells and human 293T and A549 cells infected by the NS1 delete mutant influenza A virus. Okay. Deep sequencing of the small RNA, total small RNAs from human A549 cells infected by the NS1 deletion mutant virus show that these cells accumulate abundant positive strand and negative strand influenza viral siRNA. And these influenza viral siRNAs are mostly, as you can see, 22 nucleotide long and contain mainly 1U species. So 1U referring to those small RNAs that have 5' terminal U as the first nucleotide, the U as the 5' terminal nucleotide. And that is a known property of endogenous microRNAs. Importantly, most of the sequenced 22 nucleotide viral small RNA form the canonical siRNA duplexes with 20 nucleotide base paired region in the central and 2 nucleotide overhang at both 3' end. So indicating that influenza viral siRNA are processed by DICER from the viral double-strand RNA replicative intermediate. However, the influenza viral siRNAs were undetectable in all of the 3 cell lines after infection with wild-type influenza A virus expressing the NS1 protein. So indicating that expression of NS1 protein inhibited the production of viral siRNA. So what I have shown in these lanes 3, 6, and 9, boxed by a blue line. We further sequenced small RNAs in the Argonaut complex immunoprecipitated by an antibody specific to all of the 4 human Argonaut proteins. So showing at the left of this panel are total small RNA sequence from the Argonaut complexes. So as you can see, these complexes contain mainly 21 to 23 nucleotide viral siRNA and highly enriched for 1U22 nucleotide species. Similarly, sequencing the cells infected by NS1 deletion mutant of another strain called WSN strain. They're both H1N1 serotype. Also found highly abundant 21 to 23 nucleotide viral siRNA in Argonaut complex. Note that compared to Argo-loaded population at the left, the total small RNA sequencing libraries contain less dominant 22 nucleotide viral siRNA. However, showing at the bottom panel to look for canonical siRNA duplexes with 2 nucleotide overhang. This is a -2 peak, indicating that even though the size distribution is not as obvious as for the Argo load population, these 22 nucleotide influenza viral siRNA do form these canonical siRNA duplexes. So indicating that selective loading of 21, 22 nucleotide viral siRNA into Argonaut protein. So together, our results show that in the absence of ANS1 expression, infection with influenza A virus does trigger production of highly abundant viral siRNAs, and they are loaded into Argonaut complex. We have also shown that expression of the NS1 protein inhibit the production of influenza viral siRNA. We also shown in this paper, but not shown in today, is that using the DICER knockout 293T cells kindly provided by Brian Collins' lab, we have shown that influenza viral siRNA production is DICER dependent. Unfortunately, we find these DICER knockout cells are not very useful for assaying the anti-influenza activity of the RNAi, probably because those cells do not make most of the cellular microRNAs. And it is perhaps for this reason the Jeffrey lab focused on Argo2 catalytic dead or Argo2 CD cells, mouse embryonic fibroblast cells. Okay, so just some background that human or all mammals encode 4 Argonaut protein, and only Argo2 retains RNA slicing activity, essential for RNAi by synthetic siRNA. So in the RNAi knockdown experiment, you require the Argo2 slicing activity to achieve the knockdown of your target mRNA. And also shown by Greg Hanlon's lab that mouse Argo2 with a single nucleotide mutation, a single amino acid mutation, D597A substitution, is sufficient to abolish the slicing activity. However, those MAPs, fibroblasts, were either wild type or carry instead a catalytic dead Argo2, contain similar overall microRNA population. So this is very different from the 293T DICER knockout cells. So using these wild type and mutant mouse embryonic fibroblasts, Dr. Jeffrey's lab compare the infection of NS1 delete mutant influenza A PR8 strain, showing in this slide by real-time PCR analysis. You can see the mutant virus replicated to significantly higher levels in the catalytic dead cells compared to wild-type cells, using each of the three MOI, the inoculation concentrations. So indicating that AGO2 splicing activity is antiviral against the influenza A mutant virus. And the wild-type influenza virus also replicated to significantly higher levels in the AGO2 catalytic dead cells compared to the wild-type cells. However, the increases were not as dramatic for the wild-type virus as for the NS1 deletion mutant virus shown in the slide previously. So this suggests that NS1 suppression is not complete. So the antiviral, the AGO2 splicing activity is still antiviral against the wild-type influenza virus, even though the RNA suppressor protein NS1 is present. And measuring the production of infectious influenza A virus particle by plug assay also verified the enhanced virus accumulation in AGO2 CD cells compared to wild-type cells. Notably, slicer activity of AGO2 does not appear to play a role in the interferon response because the levels of type 1 interferons and the induction of interferon-stimulated gene were similar between wild-type and AGO2 CD cells following either IAV infection or following infection or stimulation with multiple viruses or viral ligands. So indicating that slicer activity of AGO2 is antiviral, most likely by RNAi, but not by influencing the interferon response. So this slide summarizes the first set of data I have presented on the RNAi response to influenza A viral infection. I have shown that infection of monkey and human cells with influenza A virus deficient in the expression of the suppressor NS1 trigger abundant production of abundant viral siRNAs, and those siRNAs are loaded into Argonaute to mediate suppression of virus accumulation in a manner dependent on the AGO2 slicer activity. We have also provided evidence that viral, the NS1 protein of influenza A virus has viral suppressor of RNAi activity. However, many questions remain open about the function and mechanism of the mammalian RNAi response to virus infection. For example, is dicer-dependent production of viral siRNA required for the antiviral defense? Secondly, are viral siRNA produced during an active RNAi response capable of guiding sequence-specific RNA cleavage by AGO2? Does interferon signaling inhibit the production or the antiviral activity of viral siRNAs, as suggested by studies to induce RNAi using artificial long double-stranded RNA. And also, we are not very clear about the mechanism of viral suppressors of RNAi. And most importantly, is the RNAi response active in vivo? We have addressed most of these questions in a mouse model of infection by Notomira virus. So Nodamura virus, or NoV, is not a human pathogen, but it causes lethal disease in suckling mice and can be transmitted by mosquitoes, Aedes aegypti. So it is an arthropod-borne virus. Nodamura virus contains a small positive-strand RNA genome divided into 2 segments and encode only 3 functional proteins in total. The capsid protein precursor is encoded and translated from RNA2. RNA1 encodes 2 functional proteins, RNA-dependent RNA polymerase and B2, from 2 partially overlapping reading frames. So B2 and RDRP are not encoded in the same reading frame. Okay. So the RDRP is translated directly from RNA1, but B2 is translated from RNA3, which is a subgenomic RNA of RNA1 and produced after replication of RNA1. So I should also mention that because RNA1 encode RDRP, these RNA can self-replicate in the absence of RNA2. And the main reason why we have chosen Notomira virus as our model is because this virus belongs to the same alpha nodovirus genus as the Flauhas virus, a virus we have used extensively in our characterization of the fruit fly antiviral RNA response. So we know a lot about how fly cells respond to the infection by Flauhas virus. And these virus, we have shown for Flauhas virus, B2 protein is a viral suppressor of RNAi, essential for infection by inhibiting dicer processing of viral long double-stranded RNA into siRNA by sequestering the double-stranded RNA precursor. Moreover, in 2004 paper published by my lab jointly with Dr. Peter Palesi and Dr. Adolfo Garcia, we have shown that NoV replication induces ARGO2 dependent antiviral RNAi in both Drosophila cells and mosquito cells. And we have shown that NoV encoded B2 protein is an active VSR in insect cells. And most importantly, we have shown that in spite of only 19% sequence identity between the 2 B2 proteins, the conserved arginine at position 54 and 59 of the Flauhas virus and Notomira virus B2 protein, respectively, is essential for their VSR activity to suppress either dicer processing in vitro or antiviral RNAi in Drosophila cells. So remember, this single amino acid change at 59 amino acids of Notomira virus B2 will knock out its activity to suppressing lysine or RNAi in Drosophila cells. Okay. So in 2013, we reported the first evidence to show that Notomira virus infection induces and suppresses the RNAi response in circulating mice. So this slide shows that wild-type Notomira virus accumulated to very high levels in the infected circulating mice. So the fold of amplification is over 1,000. And by day 5 post-inoculation, all of the infected mice died, circulating mice died. So 100% mortality by 5 DPI. We rescued a Delta B2 mutant virus using the reverse genetic system kindly provided by Dr. Andy Ball. And this Delta B2 mutant virus contains 3 single nucleotide substitutions that prevent the translation of B2 without altering the encoded sequence of the overlapping of the encoded RDRP in the overlapping reading frame. So these 3 nucleotide mutations prevent translation of B2, but have no additional effect on the protein of the RDRP. What we find that is these Delta B2 mutant virus successfully infected the circulating mice by spreading from the inoculated abdominal cavity to the lymph tissues. So what I have shown here in a slide, real-time PCR measurement of viral RNA accumulation in the fore and hind lymph tissues. As you can see, the Delta B2 virus replicate to the peak level at day 3 post-inoculation, but then drop quickly and start from 4 DPI and 7 DPI. That basically the virus Delta B2 virus level was about 1,000-fold less compared to the wild-type virus infection. And so most importantly, all of the mice inoculated by Delta B2 virus remain healthy throughout the duration of the experiment. So indicating that loss of B2 protein abolish the virulence of the mutant virus. We further rescued another mutant virus by introducing a single amino acid R2Q mutation at position 59 of B2. So this virus we called NB2 Mutant Virus. And again, the mutation will not impact the RDRP sequence encoded in the overlapping reading frame. So as you can see, these NB2 mutant virus behave very similarly to the Delta B2 mutant virus. It replicates, spread, and accumulate to high level by day 3, but then start to drop to day 4, and day 7 is as low as the Delta B2 mutant virus. So together, these results show that the B2's VSR activity, not just the expression, the VSR activity, because the NB2 mutant virus actually express B2 protein, but is not active VSR. So indicating the VSR activity of B2 is essential for mouse infection and virulence. So sequencing the total small RNA from delta B2 mutant virus infected sucking mice revealed production of an approximately equal ratio of positive strand and negative strand viral siRNAs. These viral siRNA are predominantly 22 nucleotide long and enriched for the canonical siRNA duplexes, indicating that Notomira viral siRNA are processed by dicer from the viral dsRNA replicative intermediate. However, canonical viral siRNAs were undetectable in suckling mice infected by the wild-type Notomira virus, showing that delta B2 virus clearance in vivo was accompanied by production of highly abundant viral siRNA, and these siRNA are not visible during lethal infection by wild-type Notomira virus. So in a recent paper published jointly with Yang Li's lab in PLOS pathogen, we compared the population of viral siRNAs sequenced with or without Argonaute co-immunoprecipitation. So as you can see that these Notomira viral siRNA accumulate to very high level in Argonaute complexes, showing at the bottom panel. They are mainly 21 to 23 nucleotide long, and predominantly 22. And when you siRNA is very abundant. Mapping of the viral siRNA to the genome further revealed high densities of siRNA targeting the 5' terminal regions of RNA1 and RNA2, as well as the RNA3 encoding region of RNA1. And if you compare the pattern of the hotspot distribution, you will find the total small RNA and Argo IP RNA are very similar, so suggesting that loading into Argonaute is proportionally for the viral siRNA produced in the mice. However, we find highly abundant canonical viral siRNA in the B2 immunoprecipitates compared to the total small RNA sequenced without B2 immunoprecipitation. You can see the in the B2 complexes there are abundant viral siRNA, particularly the 22 nucleotide RNA that form these -2 kind of population. So the one that has 20 nucleotide base paired region with 2 nucleotide 3' overhang. So, however, if you look in the Argo IP population, viral siRNA are very rare, low abundant in the wild-type virus infected sucking mice. So indicating that B2 binding of duplex siRNA and but these B2 binding prevent the duplex RNA to be loaded into Argonaute. Okay, so there is a second function for the B2 shown by this set of data that B2 bind to small RNA duplex and prevent loading into RISC complex. However, this study also shows that B2 suppression of dicer processing is not complete. So there is actually some siRNAs are produced, but not loaded into RNA. So we recently investigated the genetics of RNAi response against Notomira virus in mouse embryonic fibroblasts because dicer and agar knockout mice are not viable. So we are using this system to investigate the genetic requirement of antiviral RNAi. So this slide shows the northern blot analysis of Notomira virus RNA1 self-replication in wild-type cells. And we find that wild-type cells support efficient self-replication of viral genomic RNA1 and the subsequent transcription of the subgenomic RNA3. However, both RNA1 and RNA3 were barely detectable following self-replication of delta B2 mutant RNA1, indicating an essential role for B2 in viral RNA accumulation. Notably, we find both RNA1 and RNA3 of the delta B2 mutant RNA1 accumulated to high levels in both dicer and agar2 knockout cells, shown in the middle of this panel. And as well as rescue in the cells expressing catalytic dead agar2 or agar-CD maps. So these, we further measure the virus RNA levels in those wild-type and mutant maps using real-time PCR. What you can see in the very left two bars showing the significant higher accumulation of RNA1 for the wild-type RNA1 versus the delta B2 RNA1. However, both wild-type and delta B2 RNA1 replicated to significantly high level in all of the three RNAi defective lines of cells. So indicating that B2 enhances viral RNA accumulation only by suppressing RNAi. And B2 becomes not active at all when RNAi response is abolished. Moreover, in spite of B2 expression, wild-type RNA1 replicated to significantly higher levels in RNAi defective cells than wild-type cells, indicating that compared to knocking out dicer or agar2 splicing activity, B2 suppression of RNAi is not complete, it's partial. This is a very important point. So even during wild-type B2 expression, RNAi is not completely suppressed. We also measured RISC levels expression in those wild-type and mutant cells before and after infection, and we find that B2 expression has no obvious effect on the induction of interferon-stimulated genes in either wild-type or RNAi defective lines of maps. So further supporting the conclusion that B2 enhances viral accumulation only by RNAi suppression, not by influencing the other interferon-regulated responses. We also sequenced small RNAs from wild type and dicer knockout, ARGO2 knockout, or ARGO2 CD cells after replication of delta B2 RNA1. As you can see here, there are production of viral siRNA, mainly 22 nucleotide long in the wild type cells, and highly abundant in ARGO2 knockout and ARGO2 CD cells, but not at all in dicer knockout cells. So these indicate that production of viral siRNA is dicer dependent, and ARGO2 is not required for viral siRNA production, but is required for the activity of these siRNAs. However, if we sequence the total small RNA from wild type RNA1 replicated cells, and we find only very low abundant viral siRNA in wild type and ARGO2 knockout and ARGO2 CD cells. So indicating that B2 suppresses the production of viral siRNAs. So we have recently developed an adult mouse model of infection for Notomira virus infection using RAG1 knockout strain of mice that are defective in adaptive immunity because it do not produce mature T cell or B cells, and they are intact in interferon antiviral responses. So we find that unlike the wild type C57 black 6 mice, adult mice, that are highly resistant to Notomira virus infection, the RAG1 knockout mice are highly susceptible to NoV infection, and most of the adult mice infected by NoV died. So indicating this adaptive immunity confers resistance in adult mice. However, neither of these mutant virus, delta B2 or mutant B2 virus, are able to cause any visible disease in the RAG1 knockout mice, and the infected viruses are rapidly cleared. So indicating that lethal infection of RAG1 adult mice require B2's VSI activity. And so sequencing the small RNA from the RAG1 infected mice shows that infection by either mutant virus induce abundant viral siRNA and their loading into ARGO2, but not in adult mice infected by wild type Notomira virus. So these results show that antiviral protection by RNAi is not inhibited by active signaling of interferons in RAG1 adult mice. So we further compared wild type and mutant virus infection in step 1 and step 2 double knockout mice. These mice are completely defective in signaling by all 3 types of interferons. We find that wild type Notomira virus cause lethal infection, but both of the mutant virus are defective. Defective in RNAi suppression cause no disease and are cleared very rapidly, as shown by real-time PCR analysis. So these results show that a functional B2 protein is required for lethal infection of adult mice defective in innate antiviral responses regulated by type 1, 2, and 3 interferons, further supporting the conclusion that B2 acts to suppress antiviral RNAi to promote infection, but not the interferon-regulated antiviral responses. So as expected, infection with either virus induce expression of ISGs and interferon beta genes in the wild type and RAG1 knockout mice, but not in STAT1 to double knockout mice. So these indicate that that B2 expression does not suppress the inducible interferon responses in adult mice. So the next question is whether the viral siRNA produced in the adult mice are actually active in directing RNA slicing. So we are developing a system to ask that question. So you may remember that because infected mice produce a highly overlapping set of viral siRNA, so it's very difficult, if not impossible, to map RISC slicing inside the infected cell. To overcome that problem, we synthesize artificial RNA target that contain, in this case, a T1 RNA, 42 nucleotide RNA, single-stranded, contain a central region that is perfectly best paired with a viral siRNA known to be present in agar complexes. So the predict slicing of this target RNA will produce a 22 nucleotide 5' cleaved product. So if we label the 5' end, we will be able to detect this cleavage product in a page analysis. As a negative control, we synthesize a mutant T1 RNA with only one nucleotide mutation from the T1. So this mutation would disrupt base pairing at the 10th nucleotide position, known to abolish siRNA-directed slicing. And as a positive control, we synthesize a T2 target RNA that is perfectly base paired with a microRNA 22, also accumulated in the agar complexes. So this is a mouse endogenous microRNA. So we then infected mice with delta B2 virus and use the antibody to Argonaut to prepare purified Argonaut complexes and then incubate these complexes with the T1 RNA or T2 RNA. So shown here is a page analysis showing the detection of the cleavage product of T1 after incubating with agar complexes prepared from the delta B2 virus infected RAG1 mice. So there is a very efficient cleavage. However, we did not detect cleavage using the control IgG complex immunoprecipitated from the same infected mice. We also do not see cleavage using Argonaut complexes purified from uninfected RAG1 knockout mice, all marked by the blue boxes. So we then cleavage of the mutant T1 RNA, we find using the same agar complexes purified from delta B2 virus-infected rat-1 mice, we do not see cleavage of mutant T1 that differ only by one nucleotide. So indicating that the cleavage was highly specific and induced by infection with the delta B2 mutant virus. Subsequently, we also compared the cleavage of T2 target RNA. So this is the one that targeted by microRNA. And you can see in the marked by blue, red boxes, Argonaut complexes prepared from either mock inoculated mice, or delta B2 infected mice. You see cleavage, very similar efficient cleavage. So indicating that microRNA directed cleavage is not induced or suppressed by viral infection. Okay. And these microRNA T2 cleavage was not detected if we use the control IgG, so indicating the cleavage is mediated by AGO2. Okay. So then we compared Argonaut AGO2 RNA splicing by guided by viral siRNA or microRNA from RAG1 or STAT1, STAT2 knockout mice. So as you can see, the cleavage of T2 RNA directed by microRNA 22 is similarly efficient in RAG1 mice or STAT1, STAT2 double knockout mice, whether or not they are infected or not. So microRNA cleavage is not influenced or altered by infection or not infection, or whether by a presence or active interferon signaling or complete defective interferon signaling. So in contrast, we see cleavage of the T1 RNA by viral siRNA only in virus-infected mice. Okay. However, we see cleavage of the T1 RNA in both RAG1 mice and STAT1, STAT2 knockout mice. Although the STAT1, STAT2 knockout mice, the agar complexes appear to be less efficient. So this suggests that blocking interferon signaling in STAT1, STAT2 knockout mice does not enhance production of splicing competent VSRNA AGO2 complexes. This is very different from the study using artificial long double-strand RNA to induce RNA. Okay, so we then compared RNA splicing by Argonaut complexes purified from RAG1 knockout mice or STAT1, STAT2. No, this is different. Okay. So, pardon me. So these slides actually compare agar splicing in RAG1 mice infected by Notomira virus or mutant virus. As you can see from the two lanes on the right, that infection by either mutant virus trigger very efficient splicing of T1 RNA. But the splicing of T1 RNA is very weak in a wild-type virus-infected mice, and you have to have a longer exposure to see the cleavage band. So indicating that B2 expression blocked or suppressed the production of splicing competent viral siRNA ARGO2 complexes. We also do the same comparison in step 1, step 2 knockout mice. Again, you see infection with either mutant virus triggered splicing of T1 RNA, but not in a wild-type virus infected step 1, step 2 knockout mice. So indicating that B2 expression inhibit the in vivo production of viral RNA splicing competent ARGO2 RISC in adult mice in either completely defective or intact in interferon signaling. And I think this is one of the final last data slides. This is compare expression slicing of microRNA in the Nodamura virus infected or mutant delta B2 infected mice. So showing that in contrast to antiviral slicing, microRNA directed slicing was similar, using the ARGO complexes purified from adult mice following infection with wild-type or mutant virus. So indicating that expression of the B2 does not interfere with the function of the cellular microRNA. So very in contrast to the antiviral silencing. So this is a summary that what I have shown on the study of a mice model infection by Nodamura virus. We use wild-type and RNAi defective mouse embryonic fibroblasts. We use suckling mice. We use adult mice, either intact or completely defective in interferon signaling. And so we provided 5 lines of evidence to support induction of antiviral RNAi response by Notomira viral infection. First, we show that in response to Notomira viral infection, differentiated mouse embryonic fibroblasts, suckling and adult mice produce abundant RISC-loaded viral siRNA from dicer processing of viral dsRNA replicative intermediate. And these RNA are loaded in Argonaut, direct specific slicing. So these viral siRNA loaded Argonaut are active to direct specific RNA slicing, and their activity are not affected by presence or absence of interferon signaling. And we show inhibition of RNA virus accumulation by antiviral RNAi in a pathway require both dicer-mediated production of viral siRNA and RNA splicing activity of ARGO2. And we have shown that Notomira virus encode a suppressor protein B2 essential for infection in cells and suckling mice and adult mice. There are two mechanisms to suppress RNAi, suppress dicing processing, or suppress Argonaut loading. most importantly, we find B2 only promote viral infection only by suppressing RNA, not by any other means, because B2 become inactive in RNA defective cells, and the B2 suppression is incomplete. And finally, we show that RNA response when the B2 is inactive or absent, the RNA response confer complete antiviral protection in adult mice, either intact or defective in interferon-regulated innate immunity. So this is what I have shown in my lab, but there's also additional human RNA viruses have been shown by others to encode double-strand RNA binding suppressors of RNAi, and this include the protein B2, from the piconaviridae and flareoviridae and coronaviridae and tongaviridae. And the recent studies by Zhou Xi's lab shown that VSR knockout mutant HEV71 or dengue2 induce RNAi in differentiated cell and mice. Okay, and I have to wrap up. White-type virus can also induce RNAi, and it's known for quite some time now, infection of undifferentiated cells by white-type virus or mutant virus can induce RNAi. And the recent paper published by Cateno Sosa's lab shown a novel dicer isoform in stem cells confer essential roles in antiviral RNA. So this is the last slide showing that there is an additional form of antiviral RNA immunity in mammals directed by RNA, and these response acts early, like and initiated by dicer processing of viral dsRNA replicative intermediates, similar to innate immunity. However, it has specificity mechanism, and this mechanism is different from the adaptive immunity because the specificity is programmed by viral siRNA. So finally, I'd like to thank the past and present members of my lab, and also for their contribution to the Mammalian RNAi response project we started in 2009 in the lab. And I also like to thank my collaborators. Our studies on the Nodamura virus and influenza A virus started with a collaboration with Andy Bo and Adolfo and Peter more than 16 years ago. And our collaboration with Kate Jeffrey started from a seminar visit to Boston in 2015. And also like to thank the funding provided by National Institute of Health. And finally, thank you for your attention. Bye-bye. I will be looking forward to answering questions by email.